Good morning, students. Today we will start the new chapter, that is cell cycle and cell division. In this chapter, we will discuss about how cell gets divided. What are the different types of cell division? Let us see. First of all, you see the cell cycle. Multicellular organisms grow by the addition of new cells. New cells arise by the division of pre-existing cells. That was given by Virchow in the year 1858. Duplication of DNA occurs well before cell division begins. The mode of cell division is fundamentally similar in all organisms. This shows unity of life. Phases of cell cycle. A somatic cell exists in two main states. Somatic cells means the vegetative cells, means the cells which builds the body. As you know that there are two types of cells in our body, somatic cells and reproductive cells. So first you see the states of cells. First is the interphase. That is a long undividing state of the cell, that is the interphase. And second is the mitotic phase or M phase, a short dividing state of the cell. In this phase only, the cell divides into two new daughter cells. Interphase. The cell grows by synthesizing biological molecules such as carbohydrate, lipids, protein and nucleic acids. It lasts for 10 to 30 hours. It is divided into three periods. First is the G1 phase, that is the first gap phase. Second is the synthetic phase, S phase. And second gap, that is the G2 phase. G1 and G2 phase, there is a continuous synthesis of the biological active molecules like carbohydrate, lipids, proteins, nucleic proteins, etc. And in synthetic phase only there is a synthesis of nucleic acid, means the duplication of DNA. See the pictorial view of that cell cycle. M phase, that is a mitosis, then cell division, cytokinesis, then G1 phase, S phase and G2 phase. As I told you that in G1 and G2 phase, cell prepares itself for the division, and in S phase, that is only for the replication of DNA. The amount of DNA becomes double, and once the DNA becomes double, that cell is bound to divide. That is the basis of cell division. G1 phase, gap between previous mitosis and the beginning of DNA synthesis. Carbohydrate, lipids, proteins and RNA are synthesized. No change occurs in DNA content of the cell. Muscles and nerve cells never divide again and remain in G0 phase. S phase, duplication of each chromosome by replication of new DNA molecule. Each chromosome now consists of two identical sister chromatids. A diploid cell thus becomes tetraploid, that is 4N, 2N to 4N. Synthesis of histone proteins, messenger RNAs and new nucleosomes also occur in this phase. This lasts for 6 to 8 hours. G2 phase, a gap between DNA synthesis and nuclear division. RNA transcription and protein synthesis continue. Centrioles, mitochondria and Golgi apparatus are doubled. Proteins for spindle and asterisk are synthesized. This lasts for 2 to 5 hours. Now see the mitotic phase. It is aimed at orderly distribution of the already duplicated chromosome to the daughter cells. Many structural and physiological changes occurs in the cell during mitosis. Nuclear envelope breaks down and chromatin is packed into visible chromosomes. Endoplasmic reticulum and Golgi apparatus break down into small vesicles. Microtubules dissociates into tubulin dimers. Cell activities like gene expression, protein synthesis, secretion and cell motility stops. Cell's entire attention is devoted to the process of division. Cell division occurs in three ways: amitosis, mitosis and meiosis. Amitosis, first described by Robert Remak in RBC of chick embryo, it occurs without the formation of spindle and appearance of chromosomes. The nucleus elongates and develops a constriction round its middle. The constriction gradually deepens and cuts the nucleus into two daughter nuclei. Similar constriction occur in the cytoplasm between two nuclei and divide the cell into two. Amitosis is rare. It occurs in mammalian cartilage, degenerating cells of diseased tissue and fetal membranes. Mitosis, a common method of cell division in eukaryotes, occurs in somatic cells, lasts for 30 minutes to 3 hours. Mitosis, two main steps are there: karyokinesis and cytokinesis. Karyokinesis means the division of nucleus and cytokinesis means division of cytoplasm. Karyokinesis completes in 4 phases. First is the prophase, then metaphase, then anaphase and telophase, and followed by cytokinesis. Prophase, early prophase. The cell becomes more or less rounded and cytoplasm turns more viscous. Short radiating microtubules assemble around centrioles. Two pairs of centrioles start moving to the opposite ends of the cell. Microtubules called astral rays are not in contact with centrioles. Between the asters, long microtubules assemble on one side of the nucleus called mitotic spindle. Chromosome first appears as long thin thread, gradually changes into short thick rodlets. Chromosomes are fully replicated at all points along their lengths. Prophase, the chromosomes appear condensed and the nuclear envelope is not apparent. Middle prophase, chromosomes finally assume their characteristics, form and sizes. Nuclei become smaller and finally disappear, and nuclear materials are dispersed into nucleoplasm. Nuclear envelope begins to break down into small vesicles which disperse into cytoplasm. Late prophase, chromosomes and other nuclear content are released into the cytoplasm. Centrioles pairs are pushed into the opposite ends of cells by growing spindles. Spindles and asters are together called as mitotic apparatus. Metaphase. Metaphase is the best stage for the study of chromosome. It lasts for 2 to 10 minutes. Chromosomes move to the equatorial plane of spindle, means chromosomes comes in the middle of the cell. Chromosomes should get aligned at the middle of the spindle in the form of plate called metaphase plate. The chromosomes are balanced at the metaphase plate by two chromosomal fibers that connect the sister kinetochores to the opposite poles. Anaphase. Sister chromatids of each chromosome slightly separate at the primary constriction so that their kinetochores stretch towards the opposite poles of the spindle. Chromosomes are pulled into V, J or I shape depending upon the position of kinetochores. Anaphase ends when all the chromatids reach the opposite poles. Movement of chromosomes is called anaphase A. Extension of pole is called anaphase B. Chromosomal microtubules generates the force for poleward movement of chromosomes. Telophase, long and complex phase lasts for an hour. Chromosomes at each pole unfold and long and slender. Finally, they become undistinguishable as in interphase cell. Nuclear envelope is reconstructed around each chromosome. All the envelopes fuse to form an envelope around entire set of chromosomes. Nucleolar material disperses in the cytoplasm, return to nucleolar organizer and form nucleolus. A spindle begins to disappear by depolymerization of microtubules. Asters become small till only short microtubules are left. Centrioles take up their characteristic interphase position. The chromosomes are at the poles and are becoming more diffuse. The nuclear envelope is reforming. The cytoplasm may be dividing. Karyokinesis. Two daughter nuclei formed in telophase are identical. The accuracy of karyokinesis depends upon two features: the arrangement of spindle microtubules to form two distinct poles in the cell, the connection of two chromatids of each chromosome to the opposite poles of a spindle to ensure their delivery to opposite poles. Cytokinesis, division of cell. Cell typically divide by process called furrowing or cleavage. Short spindle microtubules become structureless material called mid-body. Mid-body extend completely across the cell. Then furrows appears in the plasma membrane at the level of mid-body. With the contractile force, furrow gradually deepens. Membrane fuse. Original cytoplasm and two daughter nuclei form two independent daughter cells. New cells are half the cell size of mother cells. These enter the G1 phase of next cell cycle.